A very good afternoon and a warm welcome to all our viewers. I'm Dr. Shatil from the Clinical Engagement team here at DocPlexus. Welcome to a brand new session of the Dent-E-ssentials. The pathogenesis of periodontal disease results from the multifactorial and complex interaction between infection and host response. In addition, genetics is shown to play a role in damaging the inflammatory and immune responses of the host tissue during periodontal conditions. With the help of new technologies such as microarrays and DNA sequencing, it is possible to identify specific genetic, environmental, and behavioral factors that influence periodontitis susceptibility. To tell us more on the genetic perspective of periodontal disease, we have with us here Dr. Jasmine Crena, a renowned senior lecturer at the SRM Dental College, Chennai. Dr. Jasmine Crena is an experienced periodontist and implantologist in Chennai. She has expertise in non-surgical and surgical periodontal therapies and has several publications to her credit. In addition, Dr. Crena has a keen interest in teaching. We welcome you, Dr. Jasmine. You may proceed with the webinar. It's a pleasure to host you here today.
A very good evening to all. Thank you for the very nice introduction. So today we are going to see about a very interesting aspect of periodontal diseases. Periodontal disease from the genetic perspective is not a very new or it's been there in literature for quite a long time. It's like a big jumble where you need to take out the necessary information that is required and has to be translated. So all the information is already present. How can we take it out and get the benefit out of it is what we are right now. In this information era, with so many research going on behind the screens, how genetics would help us will be seen today in a very, very short manner. So to introduce the topic, periodontal diseases, as we all know, is a complex interplay of factors. So the factors include various factors, right from environmental factors within the oral cavity, including the plaque, the calculus, then moving on to the host responses, which include the immune response, which include the immune system function as such. Nowadays, we call it as the immune blueprint. Then moving on to the genetic aspect. Genetic aspect is the inherent aspect which is present within every cell and which is getting transmitted from one generation to another. So our body is constantly interposed with various environmental influences, but does it go into the cell and change it? It's a big question mark. Why I say this is there's some factor called as epigenetic factors. So these epigenetic factors look from the outside and they try to influence the genetic aspects. So we all say that genes never change. So that's a terminology which has been there for a long time. Nowadays, the genetic perspective has also undergone a lot of changes, and we need to understand the change which brings about a lot of information in periodontal disease, in the field of periodontal disease. So evidence indicates that the inherited variation in the DNA as such has a role which is roughly equal to that of the environment. So this determines how our periodontal health will be and how we will be affected by these environmental influences. So when we get affected because of these, so when the environment affects the genetics, it's called genetic susceptibility. What is this genetic susceptibility to do with periodontal disease? When we are susceptible, when our immune response goes down, then periodontal disease takes the upper hand. So these causative factors, which I told you, they seem to be complex because there's an interplay and they all act in a simultaneous way. So we read plaque and then it goes to gingivitis, then periodontitis. Yes, this is the pattern. But what actually happens is all this happens so suddenly and so simultaneously that we, within a blink of the eye, there are a lot of changes that happens within the cells and in the tissues. And this disease is episodic. It is non-linear in fashion. There is no, say, proper a straight line. No, it's not. It suddenly goes up. There's an episodic burst, and then suddenly it comes down. So when you do a scaling, it goes down. But does it remain the same? No. When our immune response is somewhere suffering inside, it's once again going to come back. So that is what is meant by a non-linear fashion. So periodontitis is such a type of a disease which will look maybe from the outer that it is resolved, but actually it is not resolved. It's a very, very big criminal. So, as I told you, the immune blueprint of the host thus plays a major role. So, how we maintain our immune system or how the immune system maintains us is what is playing a major role. So, in a recent paper given by Van Dyke et al., the immune blueprint has been sought or has been taught in a very, very elaborate manner to say that the immune system of the host plays a major role, and any imbalance in the immune system leads to periodontal disease. The factors that determine this immune fitness are often the factors that determine the response to the resident biofilm. So, our immune system does something to the resident biofilm that is present. So, what are the factors on that play a major role as part of the immune system? Are number one, the genetic and the epigenetic factors, because we are going to start from the tiniest. We are going to start from the cell as such, from the DNA as such, which is the most tiniest part in our human body and which determines the entire human body's function as such. So the genetic and the epigenetic factors are the first we are going to see. Then apart from that, we know very well the lifestyle factors like smoking, diet, psychosocial conditions. Apart from that, with all these things which will be made more complex diseases like diabetes, like cardiovascular diseases, asthmatic diseases, respiratory diseases. Apart from these, there are also local and dental factors which add on to it. So these local and dental factors are given a name called as stochastic factors, and they still more complicate things. So a small problem, but it's complicated by many tiny, tiny problems. That is what is periodontitis about. So a dental biofilm, when it gets influenced by these, they pose a bigger problem, which takes a lot of effort to resolve and to get healed. So currently in literature, there are about 65 genes which have been clearly suggested as being associated with periodontitis. And this has been proved in the recent genome-wide association studies that have been taught, that have been dealt with. And these have been mostly obtained from candidate gene-based approaches. So these diseases, these studies have also given us an idea about other inflammatory diseases. Like one of the very common inflammatory diseases that we have known in history is cardiovascular disease. So is there a relationship between them, and what does genetics have to do with this? Has been so the role of genetics in periodontitis has very well been clarified through the systemic diseases, out of which one is cardiovascular disease. And on this aspect, periodontists as such have played a major role in finding out what is the reason or what is the complex interplay that happens in between these two diseases. So when we talk about cardiovascular diseases, there are 4 genes or genetic loci that has been shared by both cardiovascular disease as well as periodontitis. I don't want to name them, but just you can have it in mind, like CDKN2B. It's called as andril. It is a conserved non-coding element within one of the chromosome called as CAMTA1. And apart from that, there is also a haplotype block and the VAMP8 locus. So these are some of the genes. So these shared genes, that is the genes which are shared by both the periodontitis as well as CVS, suggest that periodontitis is not causally related to atherosclerotic disease, but rather both the conditions are the sequelae, or it is the sequence of something similar. So what happens similarly is there is a similar aberrant inflammatory pathway that leads to the diseases. Both it can be either periodontitis or it can be atherosclerotic diseases. So this is a common link that has been formed. If genetics can play such a major role, and then as periodontitis, we can do something very big even in the medical field. Yes. So this emerging field of finding out a common sharing gene is an emerging field which will help us to yield new valuable information about susceptibility not only to periodontitis but also to many other medical diseases. So we need to understand what is the characteristic feature of such chronic inflammatory diseases. So for many chronic inflammatory diseases, the questions remain the same. Are the factors contributing to the onset and progression going to be the same, or what are the factors? What will be the mechanisms that keep the immune system tolerant, or how will this immune system suddenly become so exacerbated that they cause such inflammation? If we are able to answer these questions, then the treatment of periodontitis becomes very easy. So what is that trigger point is what is the need of the hour. So a person will be very good, a person would have had very good health. But suddenly one day when in your clinic he comes up with such a big inflammation, then what has triggered it? This is what we need to ponder upon. If genetics can give us this answer, yes, then genetics is the pathway for an entire new field of periodontal disease studies. So in essence, uncontrolled and unresolved inflammation needs to be studied in detail in order to find out the basis of inflammatory diseases, including periodontal disease. Because they all share common causal factors, they share common genetic factors, which is called as pleiotropy, and thereby they share common immune pathways. Thus, this knowledge has helped us in our general understanding of individual chronic inflammatory diseases, including periodontal diseases. So we have been learning periodontal diseases from the genetic perspective in populations and pedigrees. So we have come across this field of research known as genetic epidemiology through various genetic study designs. I'll just go through briefly about them. Maybe we can call them as C flats. I will explain to you what is this C flat. C stands for candidate gene approach. The F stands for familial aggregation studies. L stands for linkage analysis. A stands for association studies. T for twin studies. And S for segregation analysis. People who are interested in music can remember in this way the C flats: candidate, familial, linkage, association, twin, and segregation analysis. The candidate gene approach is a method in which a gene is mapped. So we can find out if one allele of a gene occurs more often with the disease than in the subjects without the disease. So if you have a patient X and a patient Y, you find out one particular loci, say in IL1 gene, you take one particular loci, say the 3753 position. In that, you're going to particularly look at the allele G. So if there is a variation in that gene, and if you could relate that to a cohort of patients, and you find out that such patients end up with periodontitis, then you can say that there is an association. This is how association approaches are studied, association analysis is done. So this is one of the candidate gene approach. So IL-1 is supposed to be a very commonly studied disease gene in periodontitis. And recently, the periodontal susceptibility tests chair side, which you can do and find out if a patient has periodontitis by just checking the IL-1 gene. So the next study, which I told you for genetic study design, is your familial aggregation. So you know diseases run through families. So the degree of clustering within the family, say one set of family is affected, can be estimated by comparing the number of disease cases in the relatives of the patients to the risk of the disease in the general population. In that way, you can associate and say that that disease is particularly aggregated in the family. So I had a patient recently in my clinic where he had a bleeding, particularly from his ears. So when I asked the patient about the history, he said doctors couldn't find out what is the cause, but he said that there is a familial aggregation. So he said that his grandfather had, his three brothers had, he's a family of around 7 people, 3 brothers had, and 3 brothers' children don't have, but the other brothers who did not have, their children had. So we can find out that such genetic variations occur within families, and this is called familial aggregation. Next is the linkage analysis. So as I told you now, even the cardiovascular diseases seem to be having a linkage analysis. So any molecular defects related to simple genetic diseases caused by mutation of a single gene. So when there is mutation caused in a single gene, and when the neighbor is also affected, so that genetic loci is affected. So this is called as a linkage. So when that gene is neighboring to another gene which causes another disease, this would be called as a linkage analysis. So this gene mapping strategy requires families with 1 or more members affected by the disease. You can recruit them, you can clinically and molecularly evaluate them for relatively small number of genetic markers. So for example, if a patient has periodontitis and cardiovascular disease, you can look for genes which are similar to periodontitis and cardiovascular disease if they are linked to a particular chromosome and in a particular gene where they are closer together. Then this could be a linkage analysis. Association studies, as I told you, genetic frequencies of an inherited DNA variant for a group of periodontitis cases could be statistically compared with the frequencies of the variant in a matched group of periodontally healthy control subjects. So if at all the genetic frequencies differ so greatly, then these results will be very unlikely to occur by chance. Then we can conclude that the genotype that is more common in these cases as compared with the controls would be associated with the increased disease risk. So this association studies is a very big boon for the discovery of inherited genetic variation. So if there is any genetic variation that is inherited, and if we are able to find that out, it could help us to diagnose a wide range of complex diseases like diabetes, cardiovascular disease, obesity, and even mental illnesses. Next is the twin studies. Coming to the C flats, the T, the comparison of disease occurrence in identical twins versus non-identical twins is a very powerful method for distinguishing between the effects caused by the variation in the gene versus the factors in the environment. So they will be brought up in the same environment, but does this environment affect the genetic composition? Then if you are able to find that out, twin studies will play a very major role. So if any variation among the individuals in disease susceptibility or severity is caused, so if one of the twin had succumbed to periodontitis, then very well we can say that the environment is not the one. So already the patient has been genetically affected, and although being in the same environment, one of the twin had succumbed to a disease. Okay, so that is called as twin studies. All twins, whether identical or non-identical twins, are expected to be more similar to their co-twins on average than to the unrelated members of the local population, because they have been raised in the same familial environment, they would have been having the same diet, same microbial exposure, and so on. But if the genetic variation plays an important role in determining a certain trait, then genetically identical twin pairs will be more similar to each other than the non-identical twin pairs. So this theory is because identical twins, we know that they share 100% the same genes, whereas the non-identical twin pairs share only 50% of their parents' genes on an average. So genetic epidemiologists tell this or call this as a measure of the heritability. So this heritability is based on the correlation of this disease, and this will give us the estimate of the portion of all the variations in the trait that is attributable to the inherited genetic variation. So this is about your identical twins and how your non-identical twins look like. So what idea does the identical twin studies give us? On the basis of epidemiological studies in the twins and in family studies, those with high rate of early onset periodontitis can be concluded that in younger patients, genetic contribution may be as large as 50% to the total sum of the causal factors, while in older patients, the genetic contribution to the total will be only 25%. So in a young age, they will be getting almost majority of the disease pattern. Next, the last one is a segregation analysis. Segregation will help us to distinguish different mechanisms of how the disease was transmitted through the families. So for traits in which mutation in a single gene causes the disease to develop with nearly 100% certainty in the carriers, for such persons who do not inherit the mutation are at little or they are at no risk at all. So this is another aspect. Now we finish off with the study designs. These terminologies have been very commonly used in genetic studies, and it's called autosomal dominant disease versus autosomal recessive disease. Just to give a small example, autosomal dominant disease most commonly is a Huntington disease. This occurs in the autosomal chromosomes, and it's called dominant because it's been transmitted with at least 50% probability to the next generation of the affected individuals, and thus it is found occurring across many generations of larger pedigrees. When you say autosomal recessive diseases, example is cystic fibrosis, where only 25% of the sibling will be affected, whereas the others will be just carriers. So this pattern of transmission is expected if a disease is recessive. That is, it requires the inheritance of a mutated gene copy from both parents, who themselves would have only one normal and the other could be a mutated copy, and they might be carriers. They might not be actually exhibiting the disease. So such studies will also help us to find the penetrance, genetic heterogeneity, and the inheritance pattern of the diseases. Environmental exposures are a very important modifier of disease risk. So coming to periodontal disease, such high complex combinations of multiple genetic and environmental risk factors will make the challenge of finding out or deciphering the genetic mechanisms by merely observing the transmission patterns in the families using the segregation analysis approach. That's why they are quite unfeasible. So these are some of the examples which are given. Some of the syndromes more commonly or most commonly as affecting the periodontal health would be Chiriac-Higashi syndrome. I've also given the gene which is being affected. Ehlers-Danlos syndrome, both type 1 and type 2. Then a more common syndrome, which is Papillon-Lefebvre syndrome. There are other syndromes also like Marfan syndrome, etc. So why are we discussing about genetics? We are discussing about genetics because when a treatment doesn't give us an answer, what is the reason for it? So that's why failure of resolution pathways play a major role. Resolution pathways are healing pathways that occurs after our treatment pattern, after our treatment methodologies, or after our comprehensive treatment plans. If there is a failure of this resolution pathway of inflammation, then it leads to recurrent periodontal diseases, which will have only transient healing. So recent studies have detected lower levels of such pro-resolving mediators or proteins in the whole blood of certain patients having localized aggressive periodontitis. That's why they suggest a defect which might contribute to the failure of resolution. So pro-resolving mediators are playing a major role in inflammatory resolution. So the genetic blueprint of an individual is very dependent on the type and function of the genomic variants in the genetic loci. So this genetic blueprint is a composition of or is orchestrated by multiple genes. So these multiple genes play a simultaneous role, and they play interactions, or they all interact at the same time at different locations. Not all the disease cases will have the same genetic risk variants, not necessary at all. There will be multiple genetic variations which are present at the same time. So how will we be able to find out which genetic variation has caused the disease? All these genetic variations potentially will modify the host response, and this is also not at a constant level. That is also varied. So this genetic blueprint plays a major role in understanding the complexity of periodontal disease. Not only the genetic blueprint, the influences from outside, that is the epigenetic changes that affect the DNA, causing mutations during a lifetime, further modifies the individual's susceptibility to periodontitis. So this is a multi-causality model which has been recently published in one of the papers by Van Dyke et al. In Perio 2000. So he had given us a cluster of causal factors playing a major role. So there's a pie chart containing different factors like the epigenetic factors, which is given in the blue, the lifestyle factors, which is given in orange, comorbidities, which is given in the gray, the microbial communities or the dental biofilm given in the yellow, and other factors which include two dentition related stochastic factors, which is given in the dark blue. So this could be drawn for each individual patient, for individual periodontitis patient, and the relative contribution of these 5 clusters we can analyze, and we can correlate and see. This needs to be estimated for every patient, and as such, theoretically for each patient you can do a unique pie chart which will help us to know what are the clusters of causal factors that play an important role in periodontitis. So pleiotropy between periodontitis and cardiovascular diseases, as we already studied, has been reported, and as I already told you, 4 genetic loci has been shared between coronary artery disease and periodontitis. And is it really important to know? Yes. So when there is a common causal pathway that leads to sequelae of certain medical as well as oral diseases, then why not we go back and find out what is a common pathway and give a solution to both. So these shared genes suggest that periodontitis is not causally related to atherosclerotic diseases, but rather both the conditions, as I told you, are sequelae of the similar aberrant inflammatory pathways. So when we go on to genetic studies, there have been a lot of systematic reviews, there have been a lot of literature. But the question today is can we consider these many number of cases sufficient to find out a genetic causality? Can we restrict to these many controls that we could include in original studies? Next, what are the phenotypic definitions that we can give? So today we know periodontitis is defined by the recent classification in 2017 in terms of the periodontal, the probing depth, the clinical attachment loss, the radiographic bone loss bar age. So what would be the exact phenotypic definition, and whether the investigators will be able to look into the validation of every single pilot study, whether we will be able to replicate them, All these are important. That's why systematic reviews in genetic studies have been very, very difficult. However, results of the genome-wide association studies could be more reliable because they have included almost hundreds to thousands of periodontitis patients, and they applied strict significance levels to avoid any false positive results because of the multiple testing of the genome. And mostly they could contain a validation or a replication cohort. That's why genome-wide association studies are more validated and more reliable in the present literature. One another important aspect that needs to be noted is that the human genome, the epigenetic and the genetic variations of the mutations can contribute to the microbial colonization and the infection pattern as well. This is called as the gene environmental axis. This leads to another era of genomics called as the infectogenomics. In periodontal diseases, this was introduced in 2009 by Offenbacher and Bartol. They have given us several papers and found out that genetic variants to be associated with the different levels of some bacterial species. So once again, coming to the point of epigenetics and the additional important feature which plays a modifying role or plays a major modifier in genetic blueprint of the host response to the dental biofilm in coding as well as the non-coding regions of the DNA. So studies have very prominent evidences that smoking causes inflammatory processes in the periodontal disease, and there are changes in the microbial compositions, and these changes are more commonly adjacent to the sulcular or pocket epithelial cells. So those are the most common ones which has non-keratinized epithelium. So any aberrant epigenetic modifications in these might lead to genomic DNA with functional consequences. So the interesting clinical insight from this study was that, so if we are able to do a potentially excisional periodontal surgery, then we will be able to eliminate the epigenetically modified tissues, thus yielding a normal periodontal tissue after healing. And this can presumably be able to maintain homeostasis with the dental biofilm, which is quite interesting, right? Thus, to conclude, periodontitis is a very complex chronic inflammatory disease, and it has a non-linear fashion or progression. So the host needs to exist in a symbiotic relationship with the oral microbe. So coexistence is the key. We have all learned over the period of time throughout COVID. So coexistence is the key. The factors that determine immune fitness include genetic factors, epigenetic factors, the lifestyle factors, comorbidities, local or dental factors, and factors that act randomly, and the pathobionts in a dysbiotic subgenitine biofilm. So please don't forget the pie chart. In addition to variations in the genomic sequences, epigenetic modifications of DNA can affect the genetic blueprint of the host responses. And please don't forget the genome-wide association. Studies are very much validated, and we have at least found out that 65 genes to date have been suggested to be associated with periodontitis. So with this, I conclude. Any questions could be shared.
It was absolutely wonderful listening to you. Very well presented session. We have a few questions from the audience. With your permission, let's go on to the first question. My first question to you is, how can we dentists benefit from the dissection of genetic model system for periodontitis?
Okay, that's a very interesting question. So the recent model, which is multi-causality model, which is given in a recent paper in Peru 2000 by Van Dyke et al., has shown us that these disease models could help us in a day-to-day practice to identify the causal factors. So first, we are treating a patient, but the treatment doesn't help out. So periodontitis patients, when they come, their resolution pathways need to be found out in prior, but are we really doing that? So what is the resolution pathway I'm talking about is that every inflammation has a resolution pathway. So this resolving factors needs to be identified. But how do we identify? They are blood-borne factors, and how do we identify them? What is seen clinically inside the oral cavity will be only your epithelium, your connective tissue in terms of your gingiva, as well as in terms of your periodontal ligament, in terms of your cementum. So we need to look beyond the clinical and think about certain factors. That's how the multi-causality model comes. So in our case sheet, in our day-to-day case sheet, having a periodontal risk assessment chart will definitely help us to identify where we will be able to give the patient more caution to enhance their, say, their oral hygiene practice, or their returning back, or their reviews. So whether they'll be able to keep up the reviews, all these small things can be clarified in when we have a good periodontal risk assessment chart. So this periodontal risk assessment chart seems to be a little complicated. So now this multi-causality model, which is being given, can be put up in our paper, in our very case sheet, and found out as to where we will go wrong. So when this is found out in prior, we will be able to accordingly sort out and move it in that way. Okay, so I think I've answered this question.
Well explained. Moving on to the next question. So my next question to you is, some of the papers say that specific candidate genes of susceptibility are still unknown. Would we have to wait until those genes are identified?
Okay, so this candidate-based approaches of genes have been happening in so many diseases, and periodontitis is just one small disease. Yes, it's not about waiting. It's about getting to know the disease more closer. The more better we know the disease, we will be able to treat it in a better way. So right now we have technologies, say our genome-wide association studies are itself portraying more amount of candidate genes. As of now, there have been literature which suggests that only 65 genetics genes have been identified. But our human body is quite a vast. So it's about learning the disease in a more detailed way and trying to apply that into our day-to-day practice. Waiting is one aspect. Applying it and really getting to know that patient is quite a good thing to do. So not only the candidate genes play a role. So as I told you, the gene environmental axis. So there are also other environmental factors that play a major role. So when we are able to put all these factors into our daily practice, then only we will be able to get a unified treatment plan. So that is what we have to think about. That's why point of care treatment is being nowadays propaganda in a wide platform. So we need to look into such point of care treatment strategies, and definitely yes, identifying more genes will enhance our knowledge in periodontitis, and more amount of chairside kits and chairside diagnostics could be very much improved. So artificial intelligence right now could help us in a better way. And in medical fields where we have these literature-based analysis, so such gene curation activities. So we need to curate. There are a lot of studies, but as I told you, are they really validated? Do we have the required number of patients? Do we have the required number of cases? Do we have the required number of controls? So there are a lot of bias in our studies also. So yes, genome-wide association studies are now being promoted in a larger run, and we will be able to get more candidate genes, and this will help us to have a more precision-based treatments could be possible. Customized treatments could definitely be possible. Yes. So waiting will not be wasted, I suppose.
Thank you for that elaborate answer. My third question to you is, what should be some of the latest research that we should look into or venture into for better understanding the genetic perspective?
The latest research. Okay. So artificial intelligence is one of the latest, and so how we can start about is there are a lot of projects which have been going in terms of gene curations. So these gene curations are finding out the genetic aspects from literature. So there are studies now, but how can we curate it and put it into a database? So getting a database is the new is upcoming, and it's a very big job that's being done behind us. And there are a lot of universities and institutes that have taken up this job, and it's a task to be, and but it's very complex. And only when we get to know every paper and we try to curate every information, then comes the complexity of doing that. So right now, the latest research that we can look into in terms of artificial intelligence, in terms of gene curation. So as individuals, as periodontal researchers, when we're able to contribute a little of genetic curation studies or genetic curation activities, we can produce a database that will help us to identify individual genes also, and also that will lead to more point of care chairside tests or kits in a better way.
Well explained. Moving on to the next question. What are the periodontal manifestations of immune-related disorders?
So most commonly, periodontal manifestations would be in terms of unresolving gingival enlargements. So gingival enlargements occurring in the same place, in the same site, in multiple times. So as periodontists, we will be very much perplexed. As general dentists, we will be perplexed, seeing why is it coming in the same place. So this is one of the major periodontal manifestation. So not only in a single loci, it could be even generalized. So generalized gingival enlargement is one of the very common periodontal manifestation. Apart from that, gingival bleeding. So these will never stop unless and until the underlying pathology is being identified, unless the underlying immune system malfunction is identified.
Thank you for that answer. My next question to you is, what are the periodontal manifestations of Marfan's syndrome?
Marfan syndrome, yes, as I told you, Marfan syndrome is also one of the diseases which has a periodontal manifestation, and very commonly, I think you will have a increased amount of plaque deposits. Along with that, increased amount of gingival enlargement would also be seen. So the more the accumulation of plaque, then the gingival enlargement will be more in such patients. So the same way it goes along with Chidiaki-Gashi syndrome, also the papillon-Lefevre syndrome, but invariably the age matters. So it occurs in a very young age, and these people tend to have very bad periodontal health in a very young age.
Very well answered. Moving on to the last question now. So what predictive tests can be used for periodontal disease that currently exists?
Okay. So currently existing periodontal susceptibility tests, which is I think which is not very recent, but after that we have not got still. We are still in the process of doing so. Biomarkers have been lots identified in periodontal diseases, and these biomarkers have been converted into point of care tests. So POCTs, which is nothing but point of care tests. So these will help us to identify important biomarker. But we know periodontal biomarkers are not just one. We can't just stop with one. So we need at least a different set of biomarkers. But one of the major biomarker that has been very active in the market is the PST, which is very common for the interleukin-1 gene, because interleukin-1 gene has been identified as one of the very most common inflammatory marker that has been present in periodontitis. Apart from that, even IL-6 has been very commonly found out, but this PST is more commonly for your IL-1 gene, and it's been very recently introduced in the market. Apart from that, nowadays many other biomarkers have been identified, and we can even test them. We can even check with them.
Thank you for answering that. This brings us to the end of this session. Thank you so much, Dr. Jasmine Crena, for your time and effort in putting together this interesting session. To our dear viewers, thank you so much for attending this session. Do share any more questions if you have. Until we see you again, take care and happy dogplexing. Thank you.